Dear students, this particular video speaks about the enzymes in genetic engineering. There is a need of very important tools for bringing about the production of recombinant DNA molecule. The enzymes are usually coming under five broad categories. We call them as nucleases, ligases, polymerases, DNA modifying enzymes, and topoisomerases. To begin with, let us get to know the basic and a very important type of enzyme needed for the production of a recombinant DNA molecule, an important technique in genetic engineering. And those enzymes, we call them as nucleases. So, what are nucleases? The name itself indicates these are the enzymes which act upon nucleic acid. What are the nucleic acids? It might be DNA molecule and RNA molecule. But since we speak about the manipulation of DNA, here let us speak about the nucleases always in trying to bring about the action upon the DNA or nucleic acid. It so happens when I speak about the nuclease enzymes, in this nucleases enzymes, we have two types of nuclease enzymes depending upon where they act upon in the DNA molecule. And that we call them as exonucleases and endonucleases. Now, what are the exonucleases enzymes? Exonuclease enzymes are the enzymes which are trying to bring about the hydrolysis of a terminal nucleotide from the end of a DNA molecule or a RNA molecule. When I say end, it might be on the 5' or on the 3'. For example, we have exonuclease 1 and exonuclease 2. It so happens the types of exonucleases differs upon their action, whether they're acting upon a single stranded of a double-stranded DNA molecule or whether degrading both the double-stranded DNA of double strands of a DNA molecule. For example, we have BAL31. This is an enzyme which is trying to act upon both the strands of a DNA molecule. Then we have another example, E. coli exonuclease 3. These are the enzymes which are trying to act upon only one strand of a double-stranded DNA molecule. And where are they acting upon? They're acting upon on only one terminus, that is the 3' terminus. A picture which speaks about exonuclease activity. Now we have a double-stranded DNA molecule where an exonuclease enzyme, when added upon them, what they do is that they try to bring about the splitting of the nucleotides from either end. They try to clear the nucleotides from either end, and thereby we call them as exonuclease. On the other end, we have endonuclease. So, to a double-stranded DNA molecule, when we are adding endonuclease enzyme, it so happens they try to bring about the cleaving of DNA by acting on the internal phosphodiester bonds, and thereby they are trying to give us many strands of the DNA molecule or broken strands of the DNA molecule depending upon the time and the action of enzyme on a DNA molecule. Now, this, as I told you, it is typically representing about exonuclease and examples of BAL31 and exonuclease 3. See, in BAL31, I said it is acting upon both the strands of the DNA as well as it is acting upon both the ends. But whereas exonuclease 3 enzyme, which is acting upon only one strand, and which strand it are they acting upon? They're acting upon the strand where you have the 3' end. So this is how you have the different classification of exonucleases. BAL31, it always stands for enzyme which is isolated from Anthromonas espentia. Exonuclease 3, meaning to say they have been isolated from E. coli. They were isolated from what? E. coli microorganism. The next group of enzymes coming under the nucleases are endonucleases. As told before, these are the enzymes which are trying to cut the DNA by acting upon or cleaving the internal phosphodiester bond. A good example of this is ECOR1 and 3 WAMH1, but a particular one here is nuclease S1. Nuclease S1, a enzyme, a endonuclease enzyme, which acts upon the DNA molecule or RNA molecule, but they are mainly active upon a double-stranded DNA molecule, wherein what they do is that they are trying to bring about the breaking of DNA by acting upon the internal phosphodiester bonds. The next group of enzymes which plays a pivotal role in the generation of recombinant DNA molecule, a technique of genetic engineering, are restriction enzymes, meaning to say enzymes which are trying to cut the DNA molecule at a specific position, cut the DNA molecule at a restrict position, a restriction site, and that is why we give the name as restriction endonucleases. Now, it so happens this restriction endonucleases, they're very specific in their action. They are specific, meaning to say these enzymes, they have the capacity to recognize a particular sequence, and that sequence we call them as recognition sequence, or we call them as restriction sites. Now, when I speak about recognition sequence or restriction sites, usually they are to be palindromic in nature. What do you mean by palindromic in nature? Thus are the sequences which had to be read the same from both the ends. A very good example, a conventional example is madam. So on either side or from either end, if I am reading onto this particular word, madam, okay, M-A-D-A-M, I try to repeat them in the reverse direction, that is also M-A-D-A-M. These are called as palindromic sequences. Now, the advent of recombinant DNA molecule mainly came into the picture because of the discovery of the restriction enzymes, which was done by Warren Arber, Hamilton Smith, and David Nathans, for which they were awarded Nobel Prize for the contribution. As of now, it is said that we have around more than 300 commercially available restriction endonucleases, but they have discovered around 2,500 restriction endonucleases from the species of bacteria.